is a discrete unit of hereditary information consisting of a specific nucleotide sequence in DNA. This determines the traits of an organism. Genes also carry the instructions to synthesize proteins in a cell. For eukaryotic cells, the process of protein synthesis occurs in 3 steps: transcription, RNA processing, and translation. In transcription, an RNA molecule is assembled along the DNA template strand. RNA is similar to DNA except that it contains a ribose instead of a deoxyribose sugar and has the nitrogenous base uracil instead of thymine. Since the 2 nucleic acids are written in, quote, 2 different languages, the information is simply transcribed or rewritten. A transcription factor is another protein that also binds to the DNA and participates as well, which attaches to the beginning region of the DNA called the promoter. This pries the DNA apart and untwists a section of the double helix. An enzyme participates and initiates transcription. This enzyme is called RNA polymerase. RNA polymerase moves along the DNA and pairs the RNA nucleotides to the DNA complements. Similar to DNA, RNA is also synthesized in the 5' to 3' direction. Once transcription is completed, the RNA polymerase releases the RNA and detaches from the DNA strand. In RNA processing, the RNA synthesized in transcription is modified before it leaves the nucleus. This step is specific only for eukaryotic cells. The resulting RNA from the DNA transcription is called messenger RNA or mRNA. It carries a genetic message from the DNA to the protein synthesis machinery of the cell, the ribosomes, to carry out the 3rd step. The ends of the RNA have a 5' untranslated region and a 3' untranslated region. These do not code for a polypeptide. The actual polypeptide making instructions begin with the start codon and end just before the stop codon. This portion of the RNA is called the coding segment. RNA processing begins with alteration of the ends of the RNA. A 5' cap consisting of a modified guanine nucleotide is added to the 5' end. A poly A tail made of 30 to 200 adenine nucleotides is added to the 3' end. Next, portions of the coding segment that do not actually code for proteins are removed. In translation, the ribosomes read the message from the RNA and link amino acids together to form the polypeptide. There are 3 steps of translation: initiation, elongation, and termination. In initiation, a small ribosomal unit binds to the 5' end of the mRNA molecule. The anticodon of the transfer RNA, or the tRNA, binds to the start codon. The mRNA, tRNA, and the small ribosomal unit are then brought together with the large ribosomal unit to form an initiation complex. The initiator tRNA sits at the P site. In elongation, amino acids are added one by one to the polypeptide. This occurs in the ribosome where codons pair to one another, and tRNA moves from the A site to the P site and then to the E site when it exits. The process is then repeated when a new tRNA is replaced in the A site. Elongation will continue until it reaches a stop codon, which signals termination.